Got it? All right. Same thing with that. Number 6. Right in here. Got it?
What's the lower? I do it. I can do it. I need one now. You have any clothes? I never do that. I am too. Can I do this? Anybody waiting in line to do this? People said they got burn marks.
I don't know what from.
Hold it up. Hold it up. Hold it. Just don't let go. Don't loosen up real good.
Here's one who's zipped. He knows, right? And the boys, you don't. One who's zipped. And then we'll move on. Okay, you're all set.
Oh, what's this one for?
Okay, we're going to put you up right now. Okay? All right, where's the lobster claw? Where is the lobster claw?
Right here.
There it is. Great. Can I have that, please?
I want to go.
And these things are your safety devices. This is like for mountaineering. When you get up there, you hook into that cable above your head. This is open. This is closed. That's locked. Okay?
You're dead, man. You're dead.
Open with one hand. Try it with one hand. Open it further. Twist it and says something. Open it. Now, put it in. Lock. Good.
Hey, man, I'm close, man. You gotta keep moving. That's what I was doing. You can walk across. We're gonna do this? Yes, just keep walking.
Right, you ain't not doing that.
Both hands up.
I mean,
6, 5, 4, 3, 2, 1, 0.
come on.
5, 4, 3, 2, 1, 0. That's it. Go. Man, I've been waiting for that girl. Good. Good. You gotta do one more.
I can't get it. Do you need some help?
You're getting it.
And you can get started, but you need one person per segment. So you have to cheer the guys ahead of you. Hurry up! Hurry up! Well, you can get started. You need a spotter, though.
I was helping.
Spotter? You don't look like you over here doing this. I'm a cheerleader. It's not all right, I mean it. It's a little bit after 2. 5 minutes. 5 minutes after 2. You can have open. All right. And got one more to come down, sir? She got him, Melina. Come on, there. Hey! This is the part that gets you, isn't it? You don't like this part. And it's so simple. Looks like. Did you go sit yet? Oh, I want to sit. Yeah. Hey, Mom.
Like this, not tight.
You got to unloose herself. You got
Opening it.
You got to unloose herself. You got to unloose yourself. You got to let her go off yourself.
Open it.
Getting to the end first.
Put it in. Close. See, one hand. Okay, good. All right, now. He's going to go up.
I hold up water right in front. I couldn't get the other one up there. I just knew I was going to fall never to I wouldn't make go for
Right over here. Oh, this over here. Now, you just want to be the same. Keep going. Keep going. Keep going. Would you say 6, 7, 8, 6, 7, 8, and 9?
Y'all can't go yet, Karen.
Teach them what they're doing. Right.
Dignity. Judy, one.
I'll do that. Good thing.
Okay.
Give me your bottom over a little bit more.
The rope just to let you go down, and they go and they pull you back the rest of the way.
It's a very
It's in that Bible. One of them.
What?
You guys.
Okay, we don't have to.
I'm about to tell y'all now.
Do I have to? Hey,
You don't even know me. I'm telling you, you don't even know me. Matter of fact, you don't even read.
What is it, Andy?
Nope.
Yeah, they got she can be spotted up here.
Derek, you're on video.
"45 years ago, an aggressive species of crayfish was introduced to Michigan. This new visitor, the rusty crayfish, decided it wasn't going to go home. Meanwhile, our native crayfish, such as the propinquus, began to disappear. Scientists discovered that this was because of the new visitor, the rusty crayfish. We wanted to find out if the rusty's aggressive abilities are the reason for their dominance. At the beginning of the program, we knew very little about crayfish. That meant research. But as we got ready to begin our experiment, we spent a lot of time hitting the books. We learned that crayfish have quite a complex social hierarchy. Certain crayfish are dominant, others might be considered weaklings. After the research, it was time to get our hands on some crayfish. At first, we thought building traps could be the best way to catch them. But out of 47 traps, we caught only 2 crayfish. Back to the drawing board. It was our humble leader, Mr. Webb, who suggested we go out and catch them by hand. So we went out to the river with our hip waders and nets. It proved to be more fun than the traps, as well as much more effective. Another lesson we learned is how to balance in the waders without falling over. All of us fared well except Mr. Webb, who encountered a waterfall that started to fill his waders with Rouge River water. Next, we needed a way to keep track of the specimens. Nail polish seemed like a good way to mark them. We developed a simple numbering system that we could use with the nail polish. Leuna was pretty good with nail polish. Ivan's job was to figure out which species and sex the specimens were. Jorge measured the crayfish. This was an insult to his intelligence, he thought, but somebody had to do it. Karen recorded all the information. While Patrice weighed them. We released the crayfish into the museum's reflecting pool. That didn't work too well because the goldfish ate almost all of them. Plus some of the crayfish died inside the biology lab. We asked the stream analysis team to examine the water that they had died in. They discovered that the water contained a bunch of crayfish-killing toxins. A lot of specimens had died. It looked like we were going to have to revamp our experiment, so our team decided that we could observe the crayfish right inside the lab and be just as successful. We observed them fighting for 1 week. We discovered that the rusty was indeed the most aggressive species, but it wasn't the most common. There were far more propinquus than rusties. So our conclusion was that dominance and abundance are unrelated. There are very few rusticus, but they are still the dominant crayfish. They win twice as many fights as the propinquus. But there are only half as many rusties as propinquus. We had a rough time making the experiment work. We had a great time doing it. Of course, we wouldn't have done it all without the support and teamwork of our teachers and peers.
But the Propinquus Squad stood up with this long battle, and you, the public, can help by sending donations to help the crayfish and found to get crayfish. Time out, Dad.
Thank you both for your insights, and now back to Byron.
Thank you, Tracy Pincher. And that's our special Jewish break story.
Microscopy is the study of organisms you cannot see with the unaided eye. The tools used for this work include microscopes, glass preparation slides, filters, bacteria colony counters, and other equipment. For 6 weeks, 4 intrepid young scientists explored the microscopic kingdoms for several different creatures. Creatures such as algae on the back of a turtle, protozoa on a root of a water hyacinth, bacteria living on the human body, and blue-green algae. It was discovered that living organisms survive well on the shell of a turtle. Bacteria thrive on the human body. Microorganisms live on the roots of plants. And that Kingswood Lake is receiving excessive amounts of nutrients into the water. We learn to appreciate the fascinating variety and beauty of the microscopic creatures that live with us on planet Earth.
Meanwhile, in the deep forest of the Pacific Northwest,
Most people really don't pay attention to birds. This summer, our group studied the feeding behavior of birds. The first problem we had was trying to find the best feeder that will attract a bird. Next, we observed different habitats like the ground, trees, water, and in the air. We also walked around the campus to find a spot where there were a lot of birds. We walked all over campus in areas such as the Cranbrook House, Kingswood Dorms, near certain trails, by trees, and near water. Eventually, we ended up in an area that included all these characteristics. Then we set up and waited. One problem we encountered was squirrels and chipmunks eating our seeds. Other things we got to enjoy during our 6 weeks was watching the great blue heron. The nuthatch feeding on a tree. A red-tailed hawk soaring through the sky. And seeing a Baltimore Oriole flying in the sky. We also saw the remains of a struggle between an unfortunate pigeon and a predator. In the time we had available, we found that neutral colors attracted many birds. Towards the end, our group decided to take a trip to the Detroit Zoo. We saw many things at the zoo, such as a tundra swan and a cattle egret. During this 6 weeks, we have learned to appreciate and enjoy the lifestyle of birds.
On the day of June 21st to July 31st, 1992, a group of 5 fortunate, bright students spent their summer at Cranbrook Educational Community, studying astronomy. We are Randy, Herman, Andre, Sue, and Elizabeth, and our instructor was Mr. Bass. One of the most interesting activities we did was operating the 6-inch refracting telescope. The 6-inch means a lens that is 6 inches in diameter. We needed to prepare the telescope. Here we see Andre adjusting the weights to balance the telescope. Since we spend most of our time observing during the daytime, the only star we can see is the sun. The reason the projection screen is used is because the sun can burn our eyes if we look at it directly through the telescope. Herman is recording the last hot areas of the sun, called sunspots. We also view the sun using a hydrogen alpha filter. This blocks all other colors except red. By using this filter, we are able to see prominences, which are gas eruptions on the surface of the sun. These prominences appear as tiny little flames. We also reviewed the methods used by astronomers to locate objects on the celestial sphere. By using a Macintosh computer, we are able to simulate observing through a 24-inch telescope. This is a simulation of an actual 24-inch telescope at the Crystal Lake Observatory in Florida. It shows a realistic view through the round eyepiece of the 24-inch telescope. So realistic is the view that to see the faint nighttime stars, observation has to be made under red light, as in a real observatory. Observation and data were taken on the brightness, temperature, mass, and evolution of stars. This is done using the technique of photoelectric photometry. By the end of 5 weeks of intense work, we were able to chart the birth and death of stars in an entire star cluster. It was hard work, but a good time was had by all.
Now we will have a skit performed by the 5 of us explaining astronomy and photometry.
No, you better do it. I take myself go back. I tell you, no. So you better know we still take myself go back. But that's the way I think I don't want him. Don't get one.
Botany, the study of plants and trees. A group of us had anticipated it would be boring. We refused to go into the woods as we sat there looking into the forest. Katina tried to cut our project leader's head off with a tree caliper as he yelled, "Tree snake." Katina then looked through an ebony level for the tree snake. Miss Veronica, on the other hand, took the tree caliper and used it for the true purpose, to measure the diameter. Cornelia was trying to find maple syrup as she drilled into the tree. The time for fun was over and the real job began.
These are the tools we used in our project. The tree caliper was used to measure the tree diameter in inches. This is an Abney level. The purpose was not to look for tree snakes, but to measure the tree height. This is a very rare and unique kind of tape. It was used to measure the diameter of a tree, which we see Lorena demonstrating. We used both the diameter tape and tree caliper to see which was more accurate. We concluded the diameter tape was more accurate than the tree caliper. The increment board was used to take tree samples, which determined the age of the tree by counting the growth rings. These are two of our high-tech tools, a compass, and since we couldn't afford a telescope, we invested in a Wendy's straw, which Veronica used to look through, making sure the flags were straight.
We ended up with 100 sheets that looked like this, and one big map of the forest was created. Botany turned out to be a lot of fun. In order for more students to participate in doing experiments like ours, we need them to start reconstructing the old roads and not destroying the forest to make new ones. As a conclusion of our hard work, Michael Rahidi, who comes in and helps us, has agreed to present our project to the Michigan Academy of Science in 1993.
Summer of 1992, 7 students and their team leader spent 6 weeks using their creativity and math skills to research a problem that is not thought much about: parking. It's never a problem until there is no place to park. There are many different kinds of parking structures. Some are multi-level structures. Some are underground structures where surface space is very limited. Some parking lots are very large, while others are smaller, but an extra amount of expense has been put into the architectural design so that it would blend with the surrounding area. With other lots, extensive landscaping has been added so that it will complement the surrounding area. With some lots, because of where they are located, security becomes a major factor. Yet there are still other parking lots that are just basic and some very basic. Our job was to design a parking lot that would handle Cranbrook's overflow parking problem. After the design was completed, we would build a scale model of the new parking lot. Several important parameters had to be considered in the design of the parking lot. Accurate measurements and data necessary to measure all of the different types of vehicles that will be using the parking lot. Information was recorded on the length and width of vehicles. Turning radius, and angle parking versus parallel parking, and the width of parking bays. It was time to collect all of our data and bring it back to our study area. The process of analysis was begun. We continued our research at the Detroit Public Library. We wanted to find out how others had solved similar parking problems. We were ready to begin construction of the scale model. Wood and building materials were assembled and construction begun. Slowly but surely, our design began to take shape. Learning the true meaning of teamwork, being a part of a group of 7 individuals with one common goal, to work together and to complete a successful project.
Proud people started out to boldly go where no knowledge has gone before. Their 6-week mission to analyze water ecosystems in the River Rouge watershed. Their only weapons were the 6 chemical tests: total phosphate, CO2, total hardness, silica, pH, and dissolved oxygen. Nobody ever said the mission was without risk. The team was led by a brave captain, Diane Zandy. Taught us how to use our weapons. Peter met measured the pH level, alkalinity of the water ecosystems. Damon measured water hardness, magnesium. Angela measured the total phosphate level. Krista measured the silica and carbon dioxide level. Henderson measured the dissolved oxygen level, which determines the amount of plants and animal life in an ecosystem. After being fully trained, each individual set out to fulfill their mission. Angela set out on her mission with their captain, Diane Zande. Krista also set out on her mission under the supervision of Judy Porter. Peter tested the difference between the Ruth River and Kingswood Lake. Damon compared two ponds to see if they are the same or not. Henderson wanted to determine the succession of a pond to a marsh. Along the way, we enjoyed the view of Michigan's wetlands. In the end, we came to appreciate the beauty of the environment of lakes and ponds.
Is rocketry a childhood dream or a scientific quest for height? Building a rocket takes time and craftsmanship. NASA spends multi-million dollars in state-of-the-art technology. We use Elmer's glue and cardboard body tubes. Building of the rocket took place in the Space Lab. Calculations and weighing down of the rocket required the use of different formulas and variables. Math was not the only thing that determined the flight of the rocket. Fate also played its role. When ready to launch, you need things such as a launch pad, engines, and igniters. The type of rockets used were Alpha, Hercules, and Astrocam. A solid propellant was used to thrust the rocket into the air 500 feet or more. Rocketry can be stressful, but the reward of recovery is worth it. Launching of the rocket was accomplished by pressing 2 buttons. Once the 2 buttons were pressed, there was a puff of smoke and the rocket made its way to its highest point. Once the rocket reached its highest point, the parachute was blown out. Then the hardest part began, recovery. Recovery depends on the wind factors. High wind caused the rocket to go into trees, rivers, and backyards. Low wind gets you a successful recovery. You can put any kind of payload on a rocket, even a camera. Here's the Astrocam picture seconds before impact. Rocket team to ground control. Over and out.